This video represents a little bit of a change in gears from previous videos. In the past, we looked at the history of life on Earth. We talked a little bit about evolution and its impact on culture. This is a little bit more technical. We're going to be talking about the discipline of phylogenetics. And I wanted to clear up some terminology. Most of you have had some experience to phylogeny or phylogenetics before. You may have had it in a previous class. I find often there's still gaps in understanding or even misunderstanding. So I'm going to be very clear and go over things that you might already be somewhat familiar with. Okay? So in general, the discipline of systematics includes phylogenetics, taxonomy, and classification. Sometimes you'll see people use the term phylogenetic systematics, and really they all kind of help each other out. There is quite a bit of overlap. Sometimes there's not an entirely clear boundary between one of these subdisciplines and the others. So let's just give them definitions and then realize that there may be a little bit of overlap sometime. So phylogenetics is the discipline of figuring out how organisms are related to one another. One of the central tenets of evolutionary biology is that all organisms share a common ancestor. And so in essence, we can figure out their relationships. Maybe you can think of it as kind of the family tree, or some people call it the tree of life. Taxonomy is the discipline of giving things names, right? And so we can think of an alpha taxonomy, which is species, and then we have genera, right? Each species is put into a genus, the plural is genera, and then we have families, orders, classes, and so on up the scale. And classification is the process of finding those higher level groupings, so grouping things into named groups. Now, we want our classifications to match our phylogeny, but in the past they didn't always, so that's sometimes why we make this distinction between phylogenetics and classification, okay? So just be aware of those. Again, phylogeny is finding relationships, taxonomy is naming things, and classification is putting those named things into groups. And so as you think about it, you realize there is overlap and they work hand in hand, and then all of those together are known as systematics or sometimes phylogenetic systematics. Now, phylogenetic systematics is hierarchical. And what that means is we group things into ever-expanding groups. So if we start with a species, we take several similar species and we put them all into a genus. That's a hierarchy. And then all those things in each of those different genera, maybe 3 or 4 or more genera, all get grouped together into a family and so on. And that's what we mean when it's hierarchical. There are other things, non-biological things, that are hierarchical. So the Army, for instance, is very hierarchical. Many other organizations are also. So you have a private who maybe has a supervisor named a sergeant. Sergeants then have lieutenants and so on up the line of command. It's a hierarchical arrangement also. And we want our phylogenies to be non-trivial. So let's talk a little bit about what we mean by non-trivial, because this can be a little bit of a complicated subject. So I've got a diagram picture of a closet here. Now, if I was to ask you where the socks were in this closet, you don't see any immediately, so maybe it's a little tricky to find. Where are the socks? And you'd start thinking, well, they're probably in one of the drawers. Okay, so are the socks in the top drawer or the bottom drawer? And when I teach this class in person, I ask students to raise their hand. Do you keep your socks in your top drawer or your bottom drawer? And then, usually get a number of people that are one way and a number that are the other way. And then I asked, why are some of you doing it wrong? And I do that on purpose to provoke a response, because in reality, there is no wrong place to put socks. It's really kind of trivial. If you wanted to, you could put them in, I don't know, maybe they're in one of these bins up here that's a little not very convenient, right? Maybe they're in this drawer over here. Maybe in the bottom drawer or the top drawer. Maybe they're spread across 2 drawers. I don't know. Because there's no deeper meaning to where you put your socks, right? It's probably just something that either is a personal preference or maybe something you learned from your family. So why are the shirts up here and the jackets, the sports coats down here? Is there a reason? Well, not really. It's trivial. And so classification systems can be trivial, but if we can find something deeper, then in addition to giving us a nice way to organize things, they also provide some deeper meaning. And so the system that we use to make a non-trivial system for classification is phylogeny, or relationships. We want our classifications to match and reflect the shared historical relationships of the organisms that we're classifying. Okay, so we'll show you examples and talk about that a little bit more as we move on. Now, the way that we do this is through morphology, which are physical traits of organisms, or through DNA. And notice this fits really nicely into what we talked about in a previous lecture. This really is phenotype, right, using the traits of the organism, and DNA represents the genotypes, the genetic traits, if you want. Really, so we can either use phenotype or genotype to help us discover these historical relationships about organisms. And in the next lecture, we'll look at one of the methods in a little bit of detail and give you an overview of other methods of the technical practice of how we do that. But for now, just know that we're going to use similarities, traits in either at the phenotypic level or the genotypic level to help us group organisms together. Okay, so here is a diagram of relationships among the vertebrates. And you need to start getting familiar with these diagrams. I showed you one in the very first slide also that was looking at just the tetrapod vertebrates, including some extinct lineages. So this represents the extant lineages, not all of them, but most of the major lineages of the vertebrates. Okay? So we'll come back to this one and we'll look at the tetrapod vertebrate one, which gives us a little bit more detail on that group in later things. So when we are making these relationships, do we treat all traits, whether they're morphological or based in the genetics, do we treat them all equally? And the answer is no. Hopefully you realize that, right? I could say color. Oh, let's put all the blue things in one group. Now realize that that's really kind of a trivial grouping. I could say everything that's smaller than a Chihuahua, I'll put in one group, and everything that's larger in another. And some of those may be a little bit difficult to define in a non-subjective, right? To define them objectively. But if we could define them objectively, even then, it doesn't provide any deeper meaning. It's just, well, person X decided we should do it that way. So hopefully we could come up with a widespread, even one that would apply to all organisms, way that we could classify things that doesn't have to rely on someone's opinion or someone's random decision many, many years ago. Now, some traits do not reflect historical relationships very well, and there's always going to be some disagreement and some uncertainty. That's why we need very clear methods to find phylogenies that are better than others. But there are some traits that are notoriously not very good. And then there are also other traits that are going to be good for maybe a limited use. Maybe they work well within one single genus, but don't work well outside of that genus. So for instance, color of organisms, especially across wide spectrums like this, across all vertebrates, is not a very good way to organize people or organize a species. If I put all of the blue things in one group, that wouldn't reflect anything beyond, well, they're blue, right? So instead, we try to organize them to reflect these ancient shared evolutionary connections. And so when you look at any phylogeny, really almost always they're just going to be a summary. We can't include every single group. And so we might put an image to represent one member of the group. So here I have a primate that's representing all mammals. So that's a summary. And all of the mammals we would infer would be in this one group. And if we were to zoom in here, we could probably look at a phylogeny of the mammals, right? The same thing with snakes. Snakes also belong to a group called lizards, which aren't included here, but we could have as easily put a snake here and labeled this as Squamata, which is a group that includes both lizards and snakes, and they all make a single valid group. Okay, so these are just summaries. Here we have a summary of all life on Earth, and this is an unrooted tree because when we conclude all life on Earth, we don't know how to root it because there's no closest relative. And we're going to talk about roots here in a little bit. So there are 4 terms you need to know, one which I don't have listed here. That's the root, and we'll talk about in detail later. But the other 3 I do. Terminal branches, which sometimes we will call species or taxa, are the names that we put on the end. And again, often it's just a single representative, and we might put the larger group from which we've pulled. So we put a primate on there, a monkey, but we call that terminal taxon mammals. So the terminal branches represent a group or a single species. Okay. The internal branches then have a couple of different possibilities. Sometimes they are just convenient ways. So on this phylogeny like this, these horizontal lines are just conveniently there to help us so they're spaced apart. The vertical lines going up and down can represent evolution, right? So we have evolution that has occurred to make this species different from all these other species. So the internal branches represent time passing and perhaps, not always, but perhaps evolution changing a group of organisms so it looks different from its ancestor. And then finally, the nodes represent those ancestors. And a node represents wherever lines come together. So on a triangular phylogeny like this, and you'll see both triangular and square phylogenies. On a triangular phylogeny, the node is anywhere branches come together. So that's a node, there's a node where many branches come together, right? So just recognize that. On the square ones where the branches come together, it's also true, but we have to have these horizontal lines to connect them in. So a node is right there where a vertical line goes right into the middle of a horizontal line. That is a node. And that represents the most recent common ancestor of everything above it. So that node there is the most recent common ancestor of just those two species. If I wanted to find the most recent common ancestor of all of the East African species, coded in blue here, that would be right there. That's the node. And so nodes represent the most recent common ancestors. And of course, there are more distant ancestors represented by the internal branches and then the nodes that are deeper in the phylogeny. But that's the most recent common ancestor of everything above it. And those will become important. You need to get familiar with those terms, and you need to get comfortable reading phylogenies and knowing what those nodes, branches, and eventually a root. This one doesn't have a root, but if it did, we would put it right at the bottom. It's the lowest branch coming out of the phylogeny. That's a root. Okay, now, phylogenies, when you first glance at them, will look a little bit like pedigrees, and we even called them like a tree of life, right? Some people will just use the term tree or phylogenetic tree instead of phylogeny. However, they are very different than a family tree or more technically a pedigree. And I'm going to demonstrate that right now. And we're going to do this doing a little bit of math. Now, first, I'm going to illustrate this using a pedigree of the royal houses of Europe. So you might, if you look closely, recognize some of these names if you're familiar with European history. But if not, that's fine too. But I've chosen this one for a reason. King Charles II of Spain was born in 1661. Now, there's some very interesting things going on in many royal pedigrees, but it's especially pronounced in King Charles II of Spain's pedigree. And that is why I chose him as an example. So notice, here's his dad. This is his mom. That's how you read pedigrees. We'll get a little bit more detail about reading pedigrees in a later part of the class. So father and mother. But notice something interesting about King Charles II of Spain. His mother is also his aunt's daughter, right? So typically your aunt's daughter is your cousin, right? But notice, so King Charles II, his mother was also his first cousin. Or maybe another way to put it, Philip IV of Spain married his niece. He married his sister's daughter. And then they had a son. Now notice that happens over and over and over again. So Maria Anna of Spain is not only King Charles II's grandma, also his aunt, right? And over again, we see these circles or these network-like patterns for this pedigree. That is called tocogeny. Now, in the phylogeny of organisms, we don't see tocogeny very frequently. We're going to talk about one exception to the rule here and when it does apply to species. But the vast majority of the phylogenies we look at are not going to have places where they connect back in. They'll have a branching pattern, like a tree, but no connection back in. Whereas family trees really are more like networks or webs, if you will, rather than like true trees. Okay. Now you look at King Charles II of Spain and you say, oh, poor inbred royal person. And there were some, if you look it up, there were some phenotypic consequences to this high level of inbreeding. He had lots of health issues that were probably the result of many, many generations of inbreeding. And you say, oh, poor King Charles II of Spain, he's so inbred. There's tocogeny in his family history. Well, don't get too comfortable criticizing King Charles II of Spain, because all sexually reproducing species, and your family tree also, has tocogeny in it. And although even if you don't know your family tree going back, it might not be 2, 3, 4, 5, 6 generations. You might have to go back 10 or more generations before you find an ancestor that is included on 2 different branches of your family tree. But like Thanos, it's inevitable. And I'm going to demonstrate this to you using the power of math, okay? So let me show you that. And I'm going to use Excel. If you're familiar with Excel, you might know this. You probably even maybe a little better than I am at Excel, but let me open it up just to demonstrate it. I'm going to make a simple calculation here. In the first column here, I'm going to put number of generations. I'll make these a little larger so you can read them as we go. Okay? So if I go back 1 generation, right? And nice thing about Excel, it will autofill, so we can do this. Let's do this little experiment where we go back 50 generations. So I'm going to pull it out here. It will autofill up to 50 generations. Okay? So number of generations. And in this next column, we're going to put your number, and we'll be kind of explicit here, of direct ancestors, right? People who were your great-grandfather or great-great-grandfather, somebody who you can trace directly in a direct lineage down to you, okay? So this is pretty easy. Going 1 generation back, how many ancestors do you have? Well, you think mom and dad, and we're talking genetic connections, so stepfathers, stepmothers, they don't count. But, right, you have 2 direct ancestors. If you go back 2 generations, 4, right? You have 4 grandparents. You have 8 great-grandparents, and you're going to start noticing a pattern here. Every generation you go back, the number of direct ancestors doubles, okay? And so we can represent that in a equation format, and you do that in Excel like this. Equals 2, and then I'm going to put this little notation there to that power, 2 to the first power. So that's 2. And if I do autofill down here, we're going to have 4. It automatically does the equation, which is the same as we've already put in here, 8, and then 16, 32, and so on. So if I drag this down all the way to 50 generations, it's going to calculate your number of direct ancestors you had at 50 generations. Now, before I do that and we look at that number, let's think about this. How long ago was 50 human generations? Now generations are a little bit longer. In the past, they were a little bit shorter. Let's just say 20, and so it's probably historically been close to 20 years. Maybe now it's more like 25 or 30 in some parts of the world, right? Where before people choose to reproduce. So, but between 20 and 25 years. So going back 50 generations, how long ago was that? Well, if we even think, well, let's say it's a little bit longer and say it's 50 times, sorry, let me put my equation in here, 50 times 25, right? So that's only going back 1,200 years, right? That's in historical time, still a long time ago, right? So that would be like, right, 1,200 years ago, that would be like 1,000, let's see, 1800s would be 150 years ago, 1700s would be 250. So we're talking about 8,700, right? So again, a long time ago, but still within historical times. Okay, so how many ancestors do you have if we go back to the year 8,750? Right? Let's look at it. Well, math will show us that. So I take you all the way back to 8,750. I go back to that time, and you had, now these numbers look a little funky. This is just the way that Excel does it. This is 1.12 times 10 to the 15th. So going back 50 generations, you had 1.12 times 10 to the 15 direct ancestors. Now, to the 9th power is billions, to the 12th power is trillions, to the 15th power is zillions. So you had 1.12 times 10 to the 15th, which is a little over 1 zillion with a Z, ancestors. Now you're wait a minute, the Earth was very, very crowded. There were 1 zillion people on Earth, and they all started reproducing and then eventually made me 50 generations later. Well, obviously that's not true. There weren't that many people on Earth. There were far fewer people on Earth then than there are today. And the reason is because as you go farther back, you're going to find, oh, even though you have 1,000 direct ancestors, some of those are going to be the same people once you get back 10, 12, 20 generations. And if you go all the way back to 50 generations, you're going to find all of these network-like, web-like patterns, and you're not as bad. Well, maybe you are. Hopefully you're not. You're not as bad as King Charles II of Spain, but you're going to find these interconnections. That's phylogeny. Now, this does not apply to most species. However, we're going to put one exception to the rule. So we find phylogeny in relationships between sexually reproducing species, and we also find it between species of prokaryotes. If you want to say bacteria, that's fine, between species of bacteria. And the reason we see so much tocogeny between species of bacteria is they have this way of transferring genes between organisms that are not their direct ancestors or direct descendants. And that's called horizontal gene transfer or lateral gene transfer, okay? So we'll look at that later, but this does apply to bacteria species. But for most of our phylogenies, if we're looking at sexually reproducing species, eukaryotes, then we're going to see a nice tree-like branching pattern with no tocogeny, okay? So just be aware of that important difference between pedigrees and phylogenies, okay? So we gather data. We're going to look at more detail at what we do as we infer this phylogeny. What this arrow represents is actually a very long and somewhat involved and technical process. But however we do it, we take our data and we end up with a phylogeny. Now, this phylogeny is simply a hypothesis. We have data to support it, but it may not be a very well-supported hypothesis. And if we gathered more data, or if someone else had a different data set, their set of relationships may not agree with ours. And so then we revise, we gather more data. Sometimes there's arguments back and forth. And like any hypothesis, we test it until hopefully it's fairly well established. Now, once a hypothesis gathers lots of data, lots of new people work on it, what do we call it? Right? A hypothesis is just a guess. Some guesses are better supported than others, but really it's just a guess. And once we have it well supported, we call it a theory. So really, many of our phylogenetic hypotheses are moving into the realm of where they would be considered theories. They're very, very well established. And that's an important distinction. Hopefully you got that in your introductory biology class. But remember that theory, as we use it in common language, we're often using it when really a hypothesis would be probably a better term to use. In science, a hypothesis is a guess, and a theory is a very, very well-supported body of data that there's very little doubt that that's true, right? So we have the theory of gravity, we have the theory of evolution, and then there are many other theories that have very, very well-supported data to support them, okay? So sometimes you'll see diagrams used to represent species, sometimes you'll see numbers. And remember, the nodes down here represent the common ancestor. So this would be the common ancestor of all of these red species, right? And if all of the red species share a common ancestor that's separate from all the other ancestors, that says something very interesting about that red coloration, right? And so most likely, if all of these red individuals share the same common ancestor, red coloration evolved a single time in the ancestor of all of the red flowers. If we had red flowers on many different branches and they didn't all share a single common ancestor, that's going to say something different, that red color has probably evolved multiple times, or perhaps evolved once more anciently and then was lost in some of the descendants, right? So be aware of that. I called it red, they're calling it orange, but whatever, this color evolved once. And we have a single species with a distinct shape. The clearest, most obvious explanation for that is that shape evolved one time in the recent history of this one species. Okay, so be comfortable with that. We're going to look at more and we're going to read more of them. So here's a phylogeny. Now this phylogeny on the left is the exact same as the one on the right. It's just a stylistic choice. Sometimes you'll see nodes that are highlighted with these little dots. They don't mean anything other than the person is trying to draw attention to those nodes. Okay, so we have our diagrams of species that are representing larger groups here, the lampreys. We have the branch that then connects them into the rest of the species. A root represents the most ancestral or the deepest branch of any phylogeny. Okay. Now, how do we decide where the root is going to go? And we'll talk about that in a little bit. The short answer is we use an outgroup. Okay, we try to find something that's not part of the group we're studying, but is fairly closely related to them. Now, another term that you'll see me use and that you may see in the textbook is taxon, or the plural of that is taxa. And this comes from the same root for taxonomy, and it's simply a name. It can be a species, a genus, a classification. Any of those represent taxa. So we have, as we move up our hierarchy, more and more inclusive taxa here. So we could take birds, crocodilians, turtles, snakes, mammals, amphibians, and put them all in one name group that's represented by that common ancestral node right there, and call them tetrapod vertebrates. or tetrapoda if you want to be very technical, right? We could take all of these things together, represented by this most basal node and the root, and call that vertebrates or vertebrata. Okay, so again, hopefully most of this is review, but if not, get comfortable reading these phylogenies. Now, it's important to realize that we can take a node, and if we twist the branches around that central node, we're not changing any of the relationships. So here they've represented them by shapes to kind of diagram it a little bit better. But notice they've taken the species represented here by the pyramid, the one represented by the cube, and they just simply twisted it around. But they didn't change any of the relationships. This phylogeny is exactly the same as this phylogeny because we didn't change relationships, and phylogenies only show relationships. So here is an exercise for you. I have 10 different phylogenies drawn here, and in reality you realize that the second one is only slightly different from the first one, and then each one we've made one other change. But notice the only changes we made were rotating around the nodes, and we've highlighted different nodes to show you which nodes change, but that's just a highlight. So notice in this phylogeny, relationships are exactly the same as in this one. Look at it more closely, and hopefully you can see it, but if not, look at it more and see if you can find why. This phylogeny, Roman numeral number 2 on the top, is exactly the same as Roman numeral down on the 2. It's just a different stylistic choice, but the phylogeny, the relationships, are identical. So let's demonstrate that. So 3 and 4 share a single common ancestor, and there it is, highlighted also with red on this one. 2 then shares an ancestor with that ancestor of 3 and 4. That represents that node right there. We find that exact same node down here. And then 1 connects in at the common ancestor of all of them, which the root extends from. And so there's that common ancestor of all of them. There's the same root extending. And you look at this. Even though we flip them around here, the same thing. 4 and 3, there's the common ancestor. 2 is next, and then 1. 4 and 3, 2 is next, and then 1. And no matter which of these phylogenies you pick, 4 and 3, 2, and then 1, they're all the same. Now, be careful because we could have drawn phylogenies that didn't match. So if I took this 3 here and moved it and replaced it with the 2, then we have a different phylogeny, because then 4 and 2 would share a most recent common ancestor, and 3 would be the next one, and so that would be different. But as they've drawn them here, every single one of these trees is equivalent. So I'll look at some examples. There'll probably be one on the quiz, but you need to be able to recognize when phylogenies are equivalent and when they aren't. And it's all about those shared common ancestors, those connections. Okay? So here are phylogenies that are different. So notice, and we've used both diagrams and names, but notice we have a different hypothesis in the middle than we do here on the left. The one on the left says that placental mammals, elephant being a representative of that, is most closely related to the marsupial mammals, a kangaroo is a representative. And there's the node, the common ancestor that connects them. This one's different. It says that marsupials share a most recent common ancestor with egg-laying mammals, the monotremes. And there's their common ancestor. Now notice, this hypothesis says something different about the origin of live birth versus this hypothesis. So monotremes lay eggs just like our ancient ancestors did. I don't know if you know the platypus, if you're familiar with it. There are other monotremes, a few alive, called echidnas, and they all lay eggs. So they are egg-laying mammals. Now notice the origin of live birth here occurs one time, and then both marsupials and placental mammals inherited that live birth from their common ancestor. But if these are the true sets of relationships, if this hypothesis is the real history of the organisms, then that says live birth had to evolve twice. It happened once in the ancestor of the marsupials and again in the ancestor of the placentals. Now, alternatively, you could say, well, maybe live birth evolved once down here in the ancestor of all of these mammals, but then we had a second reversion back to egg laying here. So that's a more complicated explanation than it just happened once. Okay? So let's define these groups, some of which you may be familiar with or you might have hints of from the terms we've already talked about. An outgroup is a close relative of whatever group you're studying, and it's the way we establish the root. So here, the reptiles, this lizard, serves as an outgroup for our mammals. Now, technically, we could choose a tree as an outgroup for mammals, right? That is a relative of the mammals, but it's so distant that it doesn't provide a very informative or useful way to root them. So we try to pick somewhat close relatives for our outgroup. A sister taxon is the group, it might be one branch on a phylogeny or many branches, but it's the group that is most closely related to whatever we're talking about. So in this phylogeny on the left, the sister taxon for the placental mammals is the marsupials. And we could use a marsupial also as an outgroup if we were going to do a more detailed phylogeny of the placental mammals. But on this phylogeny, the sister taxon is not the marsupials. Notice even though the marsupials are closest to the placental mammals, that's not the sister taxon. The sister taxon is the group that includes both marsupials and monotremes. Okay, so that's an important distinction. Okay, so if I'm going to ask you which is more closely related to the placental mammals, the marsupials or the monotremes, we need a phylogeny. So let's use this phylogeny in the middle first and answer that question, and then we'll see if the answer changes when we use the phylogeny on the left. What is the closest relative of the placental mammals? In this phylogeny, the closest relative is the group that includes both of these. So the echidna and the kangaroo, or any other marsupial or monotreme we wanted to include, there's their common ancestor. And that is where it connects into the placentals. So the duck-billed platypus is just as closely related to the elephant as the kangaroo is. They're both equally related because they're both part of this taxon. And we could give that taxon a name if we wanted, but now in this phylogeny, we don't have a named group for those two things together. Now, in this phylogeny, the answer to that question changes. And you need to be able to do this, right? So, what is the closest relative of the placental mammals if this is the correct hypothesis? And hopefully you say, oh, it's the marsupials, right? Kangaroo, if I had a koala bear or other marsupials on there, all of them would be equally closely related. But that is the sister taxon to the placental mammals in this phylogeny. And the duck-billed platypus is more distantly related. So in this hypothesis, elephant is more closely related to the kangaroo than the elephant is related to the monotreme. The kangaroo is more closely related to the elephant than it is to the duck-billed platypus. Okay, so just be familiar, and you need to be able to recognize and interpret these phylogenies. Okay, now our last vocabulary word on this slide is polytomy. A polytomy is when we have more than two branches coming from a single node. So notice all of the nodes we've looked at so far just have two branches. Some of them might split again later on, but there's only two branches coming out of that node. But here, this same node instead has three branches that all descend right from that same point. And that is a polytomy. For the purposes of this class, a polytomy represents uncertainty. We don't know, we don't have enough data to make a clear decision. We know all of these are mammals and belong to the same group. They all came from the same common ancestor, but we're not sure how they diverged from that common ancestor. Okay, so we have three different hypotheses. This one says placentals are most closely related to marsupials. This one says placentals are most closely related to both marsupials and monotremes, and they are marsupials and monotremes are closely related to one another. And this one says, I don't know, all of the above. We're uncertain right now. And so if we have a polytomy, we could resolve it by going out and gathering additional data. Now, sometimes polytomies can represent something other than just uncertainty, but for the purposes of this class, we'll leave it at that. Now, as we look at phylogenies and we look at classifications and compare them to our phylogenies, we need to make decisions as to whether our classifications contradict our best hypothesis about relationships. And if they contradict those hypotheses, then we need to revise our classifications. We want our classifications to match our phylogeny so that they are non-trivial. So let me give you an example. Very anciently, and this has been modified, and you may be familiar with it or not, but very anciently, these larger gray mammals were classified into a single group that we called pachyderms. So elephants, rhinoceroses, and hippos, once upon a time, were all in one single group classified together as pachyderms. And you might be familiar with that term. More commonly today, it's kind of a colloquial or an informal name for just the elephants. But anciently, it applied to elephants, rhinos, and hippopotamuses. Hippopotami, technically, but hippopotamuses sounds a little better. We recognize today that that classification is not valid. Now you say, well, why isn't it valid? They're all big and gray and heavy, but yeah, that's because they have evolved that separately. And so when we include a good sampling of mammals, we realize that elephants are not very closely related to rhinoceroses. They're related because everything's related, but very, very anciently. Hippos are not closely related to rhinos either. And so if by putting them all in one group, we are ignoring the relationships. Okay? So the only valid classification is a monophyletic classification. You need to be able to recognize monophyletic classifications. So let me give you some examples using the groups on this phylogeny, and you tell me whether it's monophyletic, whether it's valid or not. I want to take tapirs, rhinoceroses, and horses and put them all in one group. Is that monophyletic? Hopefully you say, yep, that's good. And in fact, we've already talked about that group. We gave it a name. Remember that was the Perissodactyla when we talked about the evolution of horses. Okay, now let's say I want to take pigs and ruminants and put them into one group. Is that valid? Now, when you look at it, this one's a little tricky. We haven't talked explicitly about this one. But if you look at it, that's not a valid group. And you're like, wait, they're right next to each other on the phylogeny. But yeah, still, that doesn't matter. We have to look at how they're connected. Okay? So, ruminants and pigs, we trace back. There is a common ancestor, but that's a common ancestor that ruminants share with the cetaceans and hippos. We have to go all the way back down to there before we kind of find the common ancestor between ruminants and pigs. And that common ancestor, if we try to put these in a group, we've left out one of the descendants from that common ancestor, that descendant right there that eventually diversified into these other organisms. So that is a paraphyletic grouping, and it's not valid. So if we leave out one branch from the common ancestors of the groups we're classifying, not valid. Okay? We'll come back to that in a little bit, but paraphyletic groups are not valid. So as our phylogenies get better and better and they're well established and begin to become theories, we revise any classifications that were made in the past before we had good evidence. We revise them and change them so that they match our understanding of the phylogeny. So today, pachyderm is not a valid term. It's not a valid taxa because it is polyphyletic. So let me give you one more example of polyphyletic grouping. Let's say I want to take manatees and whales and put them together and say, hey, these are the swimming mammals, right? Give them whatever name I want. Cetaceans, manatees, put them together. Now notice, to get to a common ancestor, we have to go all the way back to the root. And we've left out elephants, hyraxes, this entire group here, all of these groups. We've left them out. So if we leave out more than one group, it's polyphyletic. Now notice, when I put ruminants and pigs together, I only left out one branch, albeit a fairly major branch, but I only left out one branch. That's paraphyletic. Polyphyletic is when I leave out more than one branch by trying to put things that are very distantly related together. So, paraphyletic, polyphyletic, whatever, they're not valid. The only valid classifications are monophyletic. Okay? Now you've got a diagram over here showing you a rooted versus an unrooted tree. Sometimes you will see unrooted trees, but most of them that we're going to be putting in our phylogeny are rooted. And we do that by having an outgroup. So if this was a phylogeny of mammals, I'd put a reptile here. If it was a phylogeny of bats, right, I'd try to find, okay, what's the closest thing that's not part of the bat group that would help me to find a root. And roots may end up in different places depending on the history of the organisms and the outgroups. So notice that these phylogenies, in a rooted version, there are more versions once we add the root because it can be put in different places. Okay? But for now, just be more familiar, and we're going to mainly look at rooted phylogenies. Okay? Now, there are also different subcategories of phylogenies that you need to be familiar with. Most of the phylogenies that we've looked at so far, and most of the phylogenies we'll look at in class, are going to be cladograms. A cladogram is a phylogeny that only shows relationships. The branch lengths are not important, and usually cladograms, in fact, always, just to be consistent, the cladograms are written in one long line. All of the branches end at the same point. So here we have a cladogram showing relationships of primates. Okay? And everything ends on the same. The fact that this branch is really long and the gibbon branch is even longer doesn't mean anything. We just do it so that we can read the phylogeny easily. But it still shows all the common ancestors and all the relationships. Now, a phylogram shows all of the relationships, but in addition to that, it adds one more piece of data. And almost always phylograms are drawn in these square forms, and the lines leading to the nodes in between. So in this case, because it's phylogeny is on its side, the horizontal lines here and the horizontal lines leading to the terminal branches, those represent an amount of evolution. So the one they've highlighted here, this is looking at viruses, including the human immunodeficiency virus. But this form of HIV has evolved less than this form because of the different length of the lines. So the lines are an estimate of the amount of evolution, and that's a phylogram. Now, very rarely, and I don't even think we'll look at another one in this class, but you might see them in other classes, you might see a chronogram. A chronogram is a phylogeny, so it shows relationships. But in addition, it attempts to make branch lengths represent total amount of time that has elapsed. So remember, just because things have evolved a lot doesn't mean a lot of time has passed. We might have a very rapid rate of evolution there. It might be very slow. So that line, although it represents evolutionary change, does not represent absolute time, the amount of time that's passed. But a chronogram attempts to show that. So here we have a common ancestor, and we can look at the key here and say, oh, this may be 80, 85 million years ago. There was a common ancestor for all of the Orchidaceae, this family of plants here. Okay, so just be aware, know the definition of chronograms. We won't look at them again in this class, but you might see them in other classes. Some phylogenies have so many species included that they're really hard to read. They get difficult, and so they just have major groups highlighted. They even sometimes will swirl them around in a circle like this to make them easy to read. Just be aware of that. We won't look at these again, but that's a very many species on this phylogeny with some groups highlighted. And notice some of these highlighted groups are not valid. So on this tree, they've highlighted camelinids, one single branch, all share a common ancestor. That would be a valid classification. I think the Bromeliaceae is valid also, although we'd have to zoom in and to tell. But notice, for instance, this group, the Moraceae, is not a valid group because if they've got this green branch that is not part of it, all the brown things are Moraceae. We've got a branch over here, we've got most of them here, but then another group coming out in the middle. So that's not a valid phylogeny. We would need to reevaluate and revise this nomenclature, this taxonomy. Okay, so to end up, let's look at one last phylogeny. This is a cladogram. The branches don't represent anything except relationships. And it's a cladogram of the groups that we talked about in discussion 1.3 when we were looking at the history of life on Earth in major groups. So hopefully this will kind of reinforce some of those things. You may remember them. If not, study them a little bit more and start to get more familiar with them. Okay, so a couple things to point out. I've highlighted some nodes and given them names. That node is just an easy way to represent the entire group above it. So amniotes include all of the branches that are above it. Archosaurs include crocodiles, pterosaurs, and then this group, the dinosaurs. Sometimes, rather than labeling nodes, people will they'll put a bar above all of the species that are included in the group and they'll label it. So Dinosauria here, I've got labeled, but I don't have the node highlighted. I could have just as easily highlighted that node right there and put Dinosauria below it. So it's really convenience or whatever will fit on your diagram. So I either highlight the node, in this case the node would be right there, right, for Lepidosaurs, or I put a bar over the species that are part of that group. Okay, so just be familiar. So remember, this is tetrapod vertebrates or tetrapoda. Amphibians, we have a frog representing all the amphibians here. Here are the synapsids. I've included a tiger to represent living mammals, but remember there were other egg-laying, very reptile-like, although not included in reptiles, groups. This would be the extinct synapsids, right, like Dimetrodon. Okay, I now have the anapsids here. Now, the group Reptilia is an interesting group, and reptiles is really not used very commonly. Diapsid is actually a good stand-in for reptiles. Some people will include anapsids as part of that and call that reptiles, but more commonly, this is the anapsids are seen as a group that's closely related but not part of the reptiles. Now, reptiles, as it was originally designated, think about it. Is that a valid designation? And even if we don't look at all these extinct things, let's think about reptiles. Is it valid? And so in your head, hopefully you're saying, oh, is it monophyletic? Let's look at the phylogeny here. Now, if I ever ask you if a classification is valid, I'll always give you a phylogeny to refer to because that's what you need to do. Okay, so let's look at it. Modern reptiles include snakes and lizards, as represented here by the snake. are turtles, right? Testudines, crocodilians, and that's the original extent. There are a few other minor ones that we haven't included. If you're a herpetology fan, you might be, thinking in your head about others. But for our representative summary here, we've got snakes and lizards, turtles, and crocodiles. Is that a valid group? And obviously, when you look at it, you say no. Say, well, then let's include some of the extinct things. So let's include plesiosaurs and this branch of the lepidosaurs and mosasaurs. We'll include ichthyosaurs. And we'll call all of these things, including the dinosaurs, reptiles. Is that valid? And you realize that even if we include all of the extant and extinct things going all the way back here to the ichthyosaurs, I'll put a little circle around it, right? There we go. It's still not valid. Why? Well, it's paraphyletic. We have left out one branch, all of the birds. So just as birds are dinosaurs, if we're going to use the term reptile, we need to recognize that birds, although very highly evolved and derived, are part of the reptile group. And in fact, if we want to, right next to Diapsida, you could write in reptilia. But reptiles is used more informally now. And so sometimes people use reptiles to mean only the living diapsids, not including the birds. That's the old style classification. But today it's not used formally in a classification sense. Okay, so birds are dinosaurs, birds are diapsids, birds are part of this archosaur group, so they are included in all of these other groups. Okay, so just recognize that and realize that. And you can see why very clearly when we focus in on just the dinosaurs, why birds are classified as dinosaurs. They are more closely related to theropod dinosaurs represented by this velociraptor. By the way, that image there, let me see if I find a bigger one for you online. I find that same image. That is a much more likely, better supported by our modern fossil data and understanding, that's a much more likely representation diagram of what velociraptor would have looked like rather than the Jurassic Park version. It's slow to load here, so maybe we'll let it load. My internet connections will slow down a bit. Let's see, maybe we can zoom in here and it will be high enough resolution. Look it up if you want. But this is a better representation of what velociraptor would have actually looked like. And we talked about that, how these theropod dinosaurs had feathers, and birds are the last living remnant of the theropod dinosaurs. Are actually part of the theropod group. They're part of the dinosaur group. And so we can't have the name dinosaurs, which would include all these guys, without including birds, because